In the lab, we're interested in understanding the forces that create species and create biodiversity over time, and those forces extend back millions of years. And we're also interested in understanding how human activity in the present affects survival of species, and basically what can we do to understand changes in biodiversity into the future.
When I came in as a freshman, I was in a first-year research experience, and part of that was to match you up with a mentor. And I ended up in Dr. Berg's lab. And near the end of first semester, I was really enjoying the research, so they suggested that I apply for funding to research over the summer, and I received the Hughes grant.
I signed up for this project about 2 years ago, but I had originally started doing research with Dr. Berg when I was a freshman, so I just continued on with research because I liked it.
One highlight of my project is looking at genetic diversity and the structure of endangered freshwater mussel species, and we can use that information to look at how the species will survive into the future.
I'm studying aquatic insects that nobody studied before, and so I'm trying to build the base and work on their taxonomy, and I'm creating a guide for their identification.
So as we try to understand biodiversity, we can understand it a couple of ways. One is we can understand it by the appearance of organisms. Organisms that look the same are more likely to be closely related to each other. So we do a lot of microscopy with smaller organisms. In addition, we also tend to do a lot of DNA sequencing. The DNA sequencing lets us, instead of looking at the appearance of the organism, look at the appearance of the DNA. And the appearance of the DNA gives us a sense of how closely related organisms are.
I think the first highlight was when I first started researching the organism, because I'm working with mussels, and normally people might not think they're that interesting. But as I started to research, I realized that there's a lot of really cool things people don't know about.
One of the highlights of my research project so far has been doing field work with Dr. Berg in his lab. Traveling around is really exciting, and you get to spend a lot of time with your lab mates and develop good relationships and see the environments that the animals we're with live in.
And then we're also very interested in the geographical patterns that organisms show. And so a lot of what we do is computer mapping of organisms' distributions and looking at places where organisms overlap, where there are specific geographical and geological features that may lead to areas with high biodiversity and areas with low biodiversity.
I like being in the lab atmosphere with other people, because I realize that I learn not only a lot from my own project, but from watching other people and what they're doing too. They can teach me a lot. There's a lot more independence, like critical thinking involved, but I also like it because it provides a lot more freedom. Like you can kind of explore your own interest, and there's opportunities to pursue your curiosity.
I've published a paper actually, so that was a lot of practicing for writing, and I read a lot of literature, so I think that really has prepared me for graduate school.
After graduate, I'm currently looking at the postdoc fellowship so that I can continue my research.
My ultimate goal is to become a professor in ecology, and I would so much like to go teach in Iraq.
I hope to continue researching, so that'll probably mean getting my master's and maybe PhD in biology.
My goals after I graduate are to continue on with graduate school. I plan to find a master's program and continue the same kind of research that I do here.
The more time I spend in the lab, the more things seem like they're simple. There's usually a lot more going on.